We're going to take a look at the bacterial cell wall and the way it's put together. It's a little bit different than what you find in plants. Plants is all cellulose. The bacterial cell wall basically has two chemicals in it. One is N-acetylglucosamine. This is a glucose type material. It's very similar to cellulose. However, you can see that coming off of carbon number 2, there is an amino group. This is attached to an acetyl group, which gives it the name N-acetylglucosamine. There is another material that is also found in the bacterial cell wall, which is N-acetylmuramic acid. It's got an acid group coming off of the number 3 carbon. These together will bond to form a polymer. Instead of being a glucose polymer like starch or cellulose, it is what we call a NAG-NAM polymer. We've got the two of these. These will repeat out to make a much more extensive polymer like this. You can see in here under the NAM, we have some amino acids which can get tied into these. It makes these longer tails because the muramic acid group is able to attach to them. If we take a couple of strands of these and we turn one upside down on this, we start to get a juxtaposition of these, much the same way we would with cellulose. We get a chemical bond that forms in between the amino acid groups, the polypeptide groups coming off of the NAM, and these are often amino groups or they're amino acids, and they will cause a linkage between the two of these. This linkage between them makes a strong structure that is very resistant to coming apart. If you take this and you produce many layers of it, it becomes a very thick material that becomes much more resistant. We call this material peptidoglycan. Peptidoglycan is found in the bacterial cell wall. It is extremely important when you have a thick bacterial cell wall. That becomes important as far as what we call our Gram reaction. So we've got two different cell walls. You can see on both of these, you have a cell membrane down at the bottom. On top of that, you've got a layer of peptidoglycan. The layer of peptidoglycan in the bottom picture is thin. It's thick on the top picture. You add the crystal violet to it. The top picture gets flooded with crystal violet. It gets held in place. On the bottom picture, it's going to get a little in there. It's not going to get a whole lot because you've got another membrane on top of it. Then you add your iodine. It's going to complex it into position. You decolorize with alcohol. When you do that, it's going to destroy the outer LPS layer on the bottom one, which is going to allow the stain to leak out. On the top one, it still remains stained. And you add the counter stain, which is pink. Pink on purple leaves purple. Pink on clear leaves pink. Gram positive is purple. Gram negative is pink. General run through. 1 minute, 1 minute, less than 30 seconds, 1 minute. Watch the video and it shows you exactly how to do this.